we stopped at another sediment drift near the tip of the peninsula called Perseverance Drift and collected a casting core, a jumbo casting core 36, and that's the one I'm going to talk about, and the data I'm going to show you here comes from that. So we sampled this core for all the same chemical suites that we used in the transect assay. I'm showing just those here that appear to be, or that I think are most relevant to the question of ichthyocyte formation. So you see here down core in the diamonds is methane, dissolved methane. And you notice that this goes from a nice smooth profile to a very erratic profile. What's happening there is degassing. You can't have methane at solubilities greater than the dashed line at 0 degrees in 1 atmosphere. So when we bring these jumbo casting cores onto the deck, in a few seconds you start to hear this hissing and popping. And we pull that into the aft lab of the Palmer, and with the amount of sulfide being generated, it's almost overwhelming, but it can make your eyes water. But you really get a sense of how active this is because of the noise and buckling. Once you start pulling the plates off the side of the casting core, they pop off and the sediment wants to jump out. So that means is we've lost methane at various intervals because of the degassing. That's why you see these big jumps. But we have a nice profile for methane anywhere prior to that solubility limit. We see sulfate here. So these together define nicely the sulfate-methane transition zone, which is the condition we believe necessary for the ichthyocyte to form. Ichthyocyte being a calcium carbonate, we think you need to have sufficient calcium and carbonate concentration to supersaturate the system, so you have to be above the solubility limit, equilibrium solubility. And in order to do that, you have to generate carbonate in place. So the way that it's believed that this occurs is through anaerobic oxidation of organic matter, which does accumulate alkalinity. If you take a look at our alkalinity profile, you see a nice linear region to an inflection point, or to not an inflection, but a bend in the profile. What this argues is there's an active source of alkalinity at that same horizon where we have both methane and sulfate meeting. So nice evidence that this is a site of active anaerobic oxidation of methane. Also, the accumulating sulfide that you see suggests that this is also the active zone for this alkalinity accumulation and possible ichthyocyte formation. So other evidence that we have for the anaerobic oxidation of methane comes from isotope analysis, so del C13 of the methane produced. So this is the same geochemical profile I showed you, and now below I show the del C13 for methane across that same profile. And this is a classic distribution for these sites where anaerobic oxidation of methane is occurring. At that same depth where we see the sulfate-methane transition zone, and this, by the way, the depletion of sulfate, almost a classic linear gradient for steady-state diffusion of sulfate from the surface, ends at 80 centimeters. And at that same depth, we see the greatest depletion in methane. A paper just out this year, which I think is interesting for a number of reasons, Yoshinga have talked about this C14 ratio in the profile that we see in methane. The classic interpretation of this dip has been that the process is dominated by active methanogenesis in the regions where you see depletion in methane, and then oxidation of methane at higher depths, and then a reversal in this isotope trend. But they've reinterpreted this to being two regions on either side of a range where anaerobic oxidation of methane is occurring. Just this side is sulfate limited and this side is methane limited. Now, they've also found that this model can nicely interpret the difference between the signature in the biogenic methane rising to the SMTZ and versus the minimum value of methane that you observe. And so what I did is compare this difference in del C13s versus sulfate flux, and if we take our data and plot it on the same plot, it fits nicely with their model. Now I'll come back to this in a minute because I think it might have something to do with the regions that show ichthyocyte formation in the Antarctic Peninsula versus those that don't. So another approach we took then is just to look at the water chemistry. This is a calcium carbonate, so if you look at both calcium and carbonate concentrations, you can calculate an activity product. So you could look at ion activity products. You can also look at just the ionic concentration product. And if you have the solubility of product expressed in terms of actual ionic concentrations, you can determine whether or not the system is supersaturated or not. So I use a solubility product. This is a stoichiometric solubility product from a paper by Papadimitriou out in 2013. So this accounts for ionic strength effects already in the calculation. So if we just take a look at the product of calcium and carbonate in our system, the carbonates, by the way, we got from pH and total alkalinities that we've measured. And we see that this increases until 70 centimeters, and then 80 centimeters we see a decrease. So that drop suggests incipient precipitation of a calcium carbonate. And if we plot on this the solubility limits suggested from Papadimitriou, we see at -2 degrees, this would be saturation, and at 0 degrees, we would just be supersaturating the system in what looks like equilibration. Now, given that we're making just an estimate of carbonate concentration, that this is certainly within a ballpark value, so we think that this is pretty good evidence that we have active precipitation of ichthyocyte right at the same horizon where all the other geochemical indicators and isotopic evidence suggests anaerobic oxidation of methane is occurring. Now, one of the questions that's interesting is whenever people report ichthyocytes, they often show you these big crystals, right? And the reason for that is that's what you look for when you open up a casting core. You know, if anybody's been on a cruise where people look for these, you go in with a stick and you basically prod around till you hit something. Oh, I found one, and you pull them up, right? Now, of course, these are not precipitating as big ichthyocytes initially. So we think what's happening is they form micro-ichthyocytes initially, and then those undergo Ostwald ripening over time to form the large ichthyocytes that you discover some other time. We're interested in the rate at which that happens. I'm interested in finding some of these micro-ichthyocytes. So one of the things we're doing at the moment is trying to devise ways of separating any micro-ichthyocytes and identifying them from these sediments. I'll just add here that the idea of the rate at which sulfate is flowing and sulfate and methane are both flowing into the sulfate-methane transition zone should correlate with the degree of supersaturation you can achieve. So it may be that at these high sulfate flux sites, like ours, we have conditions that are favorable for ichthyocyte formation, but at these low sulfate-methane transition zones, there are sulfate-methane transition zones that are much more diffuse and not as compact. You may not be able to achieve the supersaturation, even though you're producing alkalinity. So we think this may be one of those parameters that drives ichthyocyte formation here. That's something we'll have to look at in the future. See, I'm running short on time, so I'm going to skip talking about the anemone here. I want to point out, I think that's a story that many of you will know already, but if you don't, you should look it up. Anemone it's a remarkable story about the organisms that mediate anaerobic oxidation of methane, the consortium between archaea and bacteria. What I did want to show you is our survey of the microbial community. And I will just mention these organisms, the anemone that mediate this process are archaea, and they're mostly closely related to methanogens, even though they're methanotrophs. And we were looking for those sequences to show up, and particularly at 80 centimeters, where we know the active oxidation of methane is occurring. In this, again, busy profile, what we see here in rank order are most abundant to least abundant taxa in our analysis of the core. And you don't see any jump up of any major group. This, by the way, are planktomycetes, not anemone. So we don't have any anemone signal. In fact, we don't find any anemone signals at all in this core, which was a shock. Now, it may be related to the fact that what we did find overwhelmingly in these cores is an enormous abundance of chloroplast sequences. These primers target all bacteria. Chloroplasts, by the way, and mitochondria are, or at least once were, bacteria. And in some cases, over 99% of our sequences came back as chloroplast sequences. So that means our deep sequencing is no longer deep. So our 15,000 reads, now we're looking at 50 to 100, or maybe 150 reads to try and characterize the community. So it's possible we're missing them. So we're going to go back and resequence, particularly target depths like 80 centimeters, with a deeper sequencing technique. So we're going to move to HiSeq, which is another platform from Illumina, which we can take as far as 1 million reads. So we should be able to push through all that chloroplast sequence to try and get to these rare sequences to get a better profile of the community. But one thing we did get out of this was the dominance of one group as you go down core, and this is a candidate phylum among the bacteria called OP9. So candidate simply means that no one's been able to grow it. But despite that fact, people have isolated, in this case, a single cell and sequenced the genome. So through genome reconstruction, we know something about this group. And one of the interesting things, again, you can't read this, but what I want to point out is through knowing what genes are present, you can predict what function may be mediated by this organism. And what we find out is that they have all the genes for hemicellulose, lysis, and fermentation. So, and the interesting thing is that the diatoms are enriched in cellulose and hemicellulose in their cell walls. So what we might be looking at is that the liberation or transformation of the organic deposition deep in the core is driven by the activity of OP9 producing acetate and ethanol, and also the fermentation product glucose as an intermediate, all of which could support either other fermentative organisms that lead to hydrogen production, or the acetate itself could be supporting acetoclastic methanogens. So methane production, all the methane we see being generated in this core might actually begin with activity mediated by this candidate class OP9. Okay, so I'm just going to say, I'm going to skip that and just say thanks, since we're short on time. These are the folks who actually did the bulk of the hard work on the ship, so I want you to know who was there. So, and with that, I'll stop. Thank you very much.
Wow. Before the obligate beer drinkers in us retired for TGIF, we have time for a few questions for Mike.
Yeah. Oh, yes, I'm sorry. Yeah.
Just one of your latest comments about not finding this chloroplast. Did you consider to profile RNA instead of DNA? Because that's usually a very
right.
way to get rid of chloroplast.
Yeah, that's a great idea. Actually, no, we haven't done that, but you're correct. We certainly can do that. That's, well, at this point, it's not a possibility, so we don't have the samples for that anymore. So we did DNA extractions of all of these. So, but next time I go to a highly enriched site that's full of chloroplasts, that's a good idea. Yeah.
Even with your existing samples, you might either changing primer or subtracting out the
Yes.
chloroplasts prior to sequencing.
You're,
Something
yes, it is. Now, so you're right, and I skipped the next steps. One of the things we're doing is going back with enemy-specific primers, and we're going to, so in addition to the deep sequencing I talked about, this is a simpler and more straightforward approach, like can we amplify them or not? And so we're going to do that. If we can't amplify them, then it's something really remarkable, so which is a little scary. So how can you have anaerobic oxidation of methane without the NME group? So, and I'm not speculating that we've got something new here, but we have to take those steps next. So, and I'd like, in terms of avoiding the chloroplast in the first place, I'd love to talk to you more about that. So if you have ideas, because we've been trying to find a way around that. So. Yes?
I'm not super familiar with microbes or sediment cores, so this might be an obvious answer, but I was just wondering, when you're looking at the different phyla that are showing up at different depths in the sediment core, for instance, the anoxic layer having the anaerobes, and is that evidence of like a community succession, or is it just they show up there because it's low oxygen deeper down?
Yeah, so that's a great question. So it's well, in this case, succession on what time scale, right? So at some point, those were fresh deposits. At that time, it was an aerobic environment. And so as these sediments eventually became buried, then you're changing the chemistry, and you do have succession on that time scale, right? But I'll add, just to play on the question of succession, it's an interesting question when we look at the transect, right? We had hoped to use time as a proxy, the distance as a proxy for time, time without ice shelf. But it's more complicated than simple distance, because there are other parameters that vary across that same scale, that same landscape. When we see this high intermediate phase of nitrification going on, is that a successional event, or is there something inherent about the chemistry of that environment, that means I should shut up, of that region that is sustainable? And is this a feature of other shelf regions in the Antarctic Peninsula? There's no other sites or studies that have been done comparably, so we're interested in seeing if that's the case. If it is a case that we're looking at a successional stage, then we may be seeing this nitrification front move towards the coastline as we move across. It may have implications for other things. I didn't talk about this, but nitrous oxide is a byproduct in the denitrification process, and even under low oxygen conditions during nitrification, it can also be produced. And it's the third most abundant greenhouse gas. How significant can that be? How broadly distributed across this region is this happening? I don't know. But there may be some significance to that. So I kind of took your question and went off on a long tangent, but sorry about that. Yes, yeah.
What's the stratigraphy of the 5-meter core? How
You need to talk to a geologist to tell you more about it.
well, does it have distinct depositional vents? Is it all just one continuous sedimentation slug, or
Well,
is there an age model?
Yeah, the strange thing about this site is it's been bioturbated. There's informal communities that have eliminated lamination. We do have the basic organic geochemistry of the TOC and the carbon isotopes of the organic matter, which Mike and I talked about. And I'll just add, what's unusual about this site, which seems to be playing into the hemicellulose model, is that there are other cores that Mike showed around the Antarctic margin, and I've looked at hundreds of them, that are equal in the content of total organic carbon. And most of that is phytodetritus, phaeocystis or diatoms. What's unusual here, which didn't dawn on us until we began looking at this cellulose model, is that these cores in the northern part of the peninsula are exceptionally rich in the total diatom abundance, which is associated with the cellulose that may be the substrate on which these
right.
particular communities
How far back
are
in time
making
are
their
we
methane.
500 centimeters. This can be.
Homogeneous diatomos.
Pardon?
Homogeneous diatomos bioturbated is the general southern
So it's all mixed up. The entire thing.
Yes.
And so there's no age model or anything like that.
Oh, we have radiocarbon dates, we have the chronology, we have the sediment accumulation rate. The accumulation rate today from lead to tin is 9 millimeters a year, almost a centimeter a year. Very high accumulation, and very, very diatom rich.
Can you carbonate it all?
Yeah.
Pardon?
Ickiites.
And
Yeah.
there's also
Some
pulsapod bivalves that we've
occasionally,
radiocarbon.
right.
So we have a 20-meter core there, which has a very good radiocarbon chronology.
Okay, so with the what we saw in the Great Australian Bight was with the H2S generation and the methane generation, tremendous amount of diagenetic alteration in the carbamates.
Mm.
I mean, they're almost, benthiformes are unrecognizable. They were overgrowths.
They're replaced or overgrowth.
Overgrowths, all kinds of crystal, it couldn't even, the micropaleontologists couldn't identify, the biostratigraphers couldn't identify what they're looking at. It was so heavily diagenetically altered. Is that going on?
Good question. We haven't looked at them, with regard to the amount of overgrowth. They look clean when we submit them for radiocarbon analysis. What's unusual here is typically the mollusks are dissolved, are not preserved in these really organic rich muds. Here in the Ickiite sites, they're there. Probably because the alkalinity is high enough to keep them in the solid phase. There's no forams. The microphone is gone. Interesting. Yeah, question? Great. Thanks again, Mike.
Oh, thank you. Thanks.